Hello, we're glad you've joined us for this live webinar, Making Polychromatic Flow Cytometry Easy After Instrument Characterization and Validation. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Thermo Fisher Scientific. Thermo Fisher is a world leader in serving science, whose mission is to enable customers to make the world healthier, cleaner, and safer. Thermo Fisher Scientific helps their customers accelerate life sciences research, solve complex analytical challenges, improve patient diagnostics, and increase laboratory productivity. For more information, please visit www.thermoscientific.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen, labeled Ask a Question, and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Grace Heinovsky, the Flow Cytometry and Imaging Facility Manager at the QIMR Berghofer Medical Research Institute in Brisbane, Australia. Grace has a strong instrumentation background and has been managing flow cytometry core facilities for more than 30 years. She came to QIMR Berghofer in 1993, having earlier worked as the flow cytometry facility manager at the Peter MacCallum Cancer Institute in Melbourne. She graduated from RMIT University with a master's in applied science. Grace is active with the Australasian Cytometry Society, organizing courses and meetings, and serves on the executive committee. She has served 2 terms as counselor for the Active International Society for Advancement of Cytometry and continues to be active with the society. Grace's complete bio is found on the LabRoots website. Grace Heinovsky will now begin her presentation.
Thank you very much, Judy, and thank you for your kind words and the opportunity to share the work we've done here at QIMR Berghofer with for instrument characterization and validation. So my role at QIMR Berghofer is to try and help facilitate good cytometry to all our internal academic users, our external academic users, and also help some of our commercial clients and contract work, as well as some of the clinical trial work we do. We have users who have got a reasonably deep understanding of flow cytometry, but then we also have those who become a little bit overwhelmed by the complexity of some of the instrumentation. We have users who prepare a panel on an analyzer, then want to do some sorting using the same panel and cannot understand why their populations may look slightly different and why one instrument gives them better separation than others. So we embarked on this set of experiments to see if we can answer some of those questions. So what did we do? Beads are the standard used for flow cytometry performance checking, and we know that these beads use different dyes to the ones that are commonly used when performing flow cytometry applications and assays. As such, we wanted to test our instrument response to the same fluorochrome that we use for our day-to-day assays and not just the dyes that are attached to the beads. Another point is that the brightness or staining index that's provided by vendors gives us an indication of how bright some of these fluorochromes may be, but that is not always the same on our instruments, on my instrument or your instrument. So we saw 61 61 CD4 antibodies with 46 fluorochromes and a number of different clones. It would have been ideal to have had the same clone for all the different fluorochromes, but we weren't able to obtain all 46 in the same clone. And initially, we started using normal healthy volunteer peripheral blood, but as you can imagine, we needed a lot of blood from one volunteer, so we could avoid any variability from different multiple donors in one experiment. After a while, we decided to start using comp beads as well. The reason is that comp beads don't change from day to day or individual to individual. The concentration is always constant, and it's a lot easier to use comp beads than large volumes of blood from different volunteers. So polychromatic flow cytometry has grown rapidly over the last 5-10 years. Many years ago, we had the organic dyes like FITC and some of the phycoproteins like PE and APC. Then along came some of the tandem dyes, Q dots, and more recently, the new Sirigen dyes. This has increased the availability of new dyes, along with new instrumentation that's able to require many more parameters simultaneously, and allowing us to measure so many more antigens simultaneously on one single cell. Having a good handle on instrument characteristics, the optical configuration can have a big impact on the ability to resolve many different sub-cell populations. Many vendors that do sell reagents with the different fluorochromes will give us an indication of their level of brightness, and some vendors will rank these dyes, as can be seen in this table. But this ranking may be different on your instrument or my instrument, either because of my optical configuration is different, or I can use different collection optics. My laser excitation power or wavelength may be different as well. Here we have some fluorochromes that are ranked in order of brightness, but also grouped in either high brightness, medium, or low. And here, again, we've got the rank from very high to very low, or also numerically from a 5 to a 1 low. So here is the list of the fluorochromes that we used in our instrument characterization, as well as the CD4 clone that was associated with that fluorochrome. There were 3 main vendors that we obtained our antibodies from, and I'll name them A, B, and C. The CD4 clone SK3 was the most common one used, followed by the RPAT4 and then a couple of the S3.5 clones. You can also see that with some of the antibodies, the same clone with the same fluorochrome was available from a number of different, or from all the vendors. For example, APC we could get for a number of the vendors. So this table shows us the optical configuration of the instruments at our institute, which we used for these experiments, and which we obtained our individual instrument characteristics on. You can see that some of the instruments have got identical optical configurations, such as the Fortessa 1 and 2 are identical. The Fortessa 4 and 5 are also identical, so they have the same lasers, the same dichroics, the same collection filters, whereas the other ones may have slight variation with some of their bandpass filters and also dichroics. I haven't listed the dichroic filters or the searing optics here, but they may also differ slightly from instrument to instrument. Okay, so antibody titer. All antibodies are titered for both the normal healthy volunteer or the peripheral blood, but we also did titers, compared titers using the comp beads. We expressed the titer as protein in micrograms per mil. We wanted to make sure that the same amount of antibody is present from all the antibodies that we tested, and the only difference was the fluorochrome that we were using. And we wanted to also make sure that, once you have a titer with your peripheral blood, that's a lot easier to obtain. But with comp beads, we really wanted to make sure that the amount of protein, because of all the binding sites on comp beads, we wanted to make sure that the protein between all the fluorochromes was the same for the beads. So the staining index, what is the staining index and what does the staining index tell us? It lets us know how well resolved a population is from the background or from the negative population. The brighter the signal above the background or the unstained in a sample, the higher the staining index. So for all the data required for each antibody from each titer, we calculated the staining index. You calculate the mean fluorescence intensity, or the MFI, of the positive cell population, the MFI of the negative cell population, then calculate the standard deviation of the negative population, and then you use the formula where you subtract the negative MFI from the positive MFI and divide it by 2 times the standard deviation of the negative population. As the negative population expands, or the staining index, the standard deviation will also increase, which will then have an effect on the staining index. The staining index allows us to determine how well resolved our CD4 population is from the background or the negative populations. Here's an example of some of the staining index results we got and what we considered was the optimal titer. So we did this for all the fluorochromes. I've also plotted the positive median fluorescence and the negative median fluorescence, and then in red we can see the staining index. You can also see that sometimes if a population, if you've got the highest mean fluorescence intensity on a positive population, that doesn't necessarily mean that higher titer or that concentration is going to give you the best staining index, because sometimes you will get an increase in the negative, which will have an effect on the total staining index, or that final staining index number. So it's about getting the best separation between your negative cells and your positive cells. Okay, so we then plotted all our different staining indexes for the different antibody titers. And what we can see on this plot is what we were looking for. We've got 3 different vendors with the same PE-CD4 clone. The results are very, very similar on each instrument. And these 2 instruments are identical. Same lasers, same optical configuration, but the staining index is markedly different on both of them. And this is what we hope to achieve by performing these experiments and seeing which instrument behaves differently, or how they behave differently with the different fluorochromes. We also did some voltration experiments where we altered the voltage for each detector just to see what was the best voltage for, you know, the different fluorochromes, and looked at the range where you do get a better, you know, signal between your negative and your positive cells. So the results. Here we've got one of our, the response from one of our instruments, one of our Fortesses. It has 5 lasers, and we're able to configure the instrument to acquire data from all the fluorochromes we tested. So we can see here that the 2 fluorochromes that give us the best staining index are from 2 different vendors. They're excited by the same laser, collected from the same detector, and also these 2 fluorochromes have got similar spectral properties. So what this is indicating is that the detectors that are picking up those fluorochromes are very sensitive to that the spectrum, and also those 2 fluorochromes are reasonably bright as well. Go back. So here we've got the 4 instruments. The 2 instruments, the 2 top instruments are identical in their optical configuration, and the bottom 2 are also identical with their optical configuration. The 2 on the left are older instruments. The FH04 and the F4A are older instruments, and the 2 on the right have been purchased in the, you know, more recently. The 2 on the left were very popular. They were used, you know, they're usually quite booked out, and the users like the configuration, they like to run their experiments, and they said, well, we need instruments that are exactly the same. So we purchased additional instruments with the same configuration to keep our users happy, and we can see that the older instruments don't have as good a response as the newer ones, and later on in this presentation I will try and address why this is, or why I think, you know, this is. Here we have one of our cell sorters, and you can see that the response to most of the fluorochromes is very good, but the response to the fluorochromes excited by the 561 laser are very good. So just before we perform these instrument performance characteristics, we had a few issues with the instrument, and the 561 laser died. So we got a brand new laser, all the fibers, everything had been replaced, and everything had been realigned, which could be another reason why the yellow-green is giving us much superior signals to the rest of the fluorochromes. So this is the stream-in air cell sorter. It's our MoFlow, and again, we've got an excellent response to the yellow-green fluorochromes. One thing that we did do for this instrument, when the new fluorescent protein dyes became popular, we purchased a reasonably high-powered laser to excite some of these fluorescent proteins, such as M-cherry. So the power on this from this laser is a lot higher than the power on the other lasers on the instrument. Also can explain why we're getting such a good response. So do the same fluorochromes have better response on our instruments from the different vendors? No, not really. They're all pretty much the same. We compared the same CD4 clone with the same fluorochrome from different vendors and found that they pretty much gave similar results on all our different instruments. So we've got the APC, FITC, PSI7 and PE, and all the instruments and the responses. So they all were where you've got a better response from one fluorochrome, you also got a reasonably good response from the other vendors as well. Comparisons between different fluorochromes, but where you're using the same detector. So when you compare different fluorochromes from different vendors, there are differences, and this could be due to the actual brightness of the dye, but it could also be due to the specific collection optics of that instrument. You know, some filters may be better suited for a particular fluorochrome than another, and we can't, constantly be changing our collection filters to accommodate from one fluorochrome, from one experiment to another. And you can see that, some of the emission spectra are reasonably close, but they're not all identical. So in an ideal flow cytometry world, our dyes would emit a very narrow spectral band with no or very little spillover signal into different detectors. The reality is fluorochromes emit, into a whole lot of other detectors where you don't want them to, either to adjacent detectors, but also to other detectors where you've got different excitation wavelengths. Sometimes this spillover can be quite significant, and this will make multicolour or polychromatic cytometry and panel design difficult. When there's spillover of this sort of fluorescence into multiple detectors, it will have an effect of spreading the nominal negative cell distribution, and this can have a huge impact on the ability to resolve some populations, especially those that have got a very low antigen expression. So what we did from after we obtained all these instrument characteristics, we used a feature in FlowJo that is able to calculate the spillover spreading matrix between the different fluorochrome combinations. The software does make it easy to recognise which fluorochrome combination should best be avoided, as they do have a very, very high level of spillover into each other. So if you cannot avoid using some of these combinations, you should try and assign the dim receptors to the bright dyes that do have a minimal, spillover. And in the table, you can see some examples where you've got, our PE-Sci5 and a BB700. There's, that value is quite high. And, if you had two dim populations on either of those fluorochromes, you may not be able to resolve that population very well. So here we have another table where we're able to summarize all the spillover, and you can see, which fluorochromes are going to give you quite high, spillover of that fluorochrome into the other detectors. And you can actually calculate the total spread from each dye contributes, and it kind of lets you know whether that dye is going to, work well or play well with, some of the other dyes, which is very useful when you're designing a new panel, and also when you're adding, one or two extra fluorochromes or markers to an already prepared panel. So down the bottom here, you can also calculate the total spread. Detectors that have got very low values will be most sensitive for detecting when you've got, all the other colours used. And this is very important when you're trying to resolve very low dim populations. And again, those dim markers, try and put them on the brightest dye with the least spread to try and, have some to guarantee success. So I'm going to try and explain what I think some of the reasons are for the differences in staining index. So some of the things that may have an effect on the staining index and how well we're able to resolve our cells of interest is the laser power. On one of our analyzers, we're able to change the laser power, and we ran some 8-peak rainbow beads just to see the effect of the laser power and the ability to resolve all 8 peaks. As we increase the laser power, we're able to resolve some of the dimmer bead populations. We can see that, here we've collected we've used two different collection filters for these beads, and you can see that no matter how high you turn up the laser, some of the dim populations are not as well resolved in the higher wavelength, the 670-30 collection. And that's because the dye emission doesn't peak around that area. It sort of peaks at the lower wavelengths. So here we also looked at another analyzer where we're again also able to change the laser power and the effect it did have on resolving 8-peak beads. So laser power is important. Maybe not so for some of the bright expressing beads or bright expressing dyes, but where you do have very low antigens or very dim populations, laser power can help improve the resolution. And again, this is another representation of how the laser power does have an effect on resolving the low bead populations. So optical filters, bandpass filters, dichroic filters, they're made of different layers and coatings that allow you to collect the different wavelengths of interest. Earlier in the presentation, I showed a couple of instruments which in theory should have been identical, having the same optical layout, but we saw that with some of these, we knew that some of the newer instruments gave us a better staining index than the older ones. And one reason for this is that the bandpass filters in the older instruments have degraded. So environmental deterioration due to either moisture, getting into some of the layers on the bandpass filters will have a big impact on how well these filters behave. Brisbane's in Australia in the subtropics, and our summers are hot and very, very humid. And no matter how hard we try and control the humidity and keep, the temperatures at a constant level, there is still a lot of moisture in the air, and I'm pretty sure that a lot of the our bandpass filters do degrade just because, there is a lot of moisture in the air. And something that you can try out there is if you take your filters out of your instrument, if you don't think they're, performing well, and just put them up against some light, you can actually start to see some degradation. And when you do start to see that, it's time to swap the filters. There are different vendors that do supply filters that, as the quality of the filter goes up, so does the price. So in this slide, we can see, the different effects of, or the effects that filters can have on how well we're able to resolve populations. Here we've run some 8-peak rainbow beads and swapped only the bandpass filter collecting the signal. Everything else was the same, the dichromatics, the laser power. We used the same bandpass filter of collecting 530/30 wavelengths. Some of the new filters may not be as good as, one would think. Some of the older ones you can see are so bad that you can't even resolve, some of the higher peaks well. So, bandpass filters do play a big part in how well you're able to, or how bright a signal you're getting on your instrument. So, if your instruments are quite a few years old, it's really worth sitting down and checking, have your filters degraded? Do they need to be replaced? And if so, when you do replace them, you'll find that there's a marked difference on how well your instrument will start to perform. So photodetectors or photomultiplier tubes convert the photon of light being emitted from our fluorochromes and convert these into an electronic signal that we can then measure and display in flow cytometry. As the number of photons hitting the detector increases, the current, the resulting current also increases. Photomultiplier tubes have got different responses to different wavelengths, and the manufacturers will select photomultiplier tubes for a range of wavelengths to be able to detect within the visible range. Some photomultiplier tubes have got much better responses to a particular wavelength than others, and sometimes they may not be placed in the best, the most ideal spot for a very or for its most suitable wavelength. Years ago, with the instruments I had, I used to remove all the photomultiplier tubes from an instrument and then test each one individually for different wavelength responses, and I would then place them on my machine in positions that were best suited to collect that particular wavelength. The newer instruments these days make that a little bit more difficult. Everything's sort of bolted down and locked in the instrument. Some of the cell sorters still allow you to do that, but most of the analyzers, it's not so easy to do anymore. And something else that people should be aware of, if you do have very, very bright dyes that where you've got a very high signal going to that detector, at that higher detection rate, some of the photomultiplier tubes may not respond in a linear fashion anymore, and they do start to waver when the incident light is very, very high. So the Q, what is your Q? So the Q value gives you an indication of how efficient a detector is able to convert that light signal or that fluorescent signal to an electronic signal or a photoelectron. If you've got a cell that perhaps has 10 fluorescent molecules being emitted that hit the detector and the conversion is 5 photoelectrons, then the Q value would be we've got a 50% conversion. The average conversion in most flow cytometers, which is very good, is around maybe 20 to 30%. So not every single fluorescent molecule gets converted into a photoelectron. So what kind of things affect the Q value? As I showed earlier, laser power, but also laser alignment. You may have a laser with really good power, but if it's not aligned properly and it's not focused properly, you're not going to get the most optimal signal. Cleanliness, you really need to make sure that you look after your instrument. You've got to have a clean flow cell or nozzle. Make sure all your steering or your objectives are clean, your lenses are clean. If you've got neutral density filters in front of some of your detectors, that will also decrease the Q value. As I said before, PMT sensitivity. Some detectors are, give a better response to some wavelengths, and some are less sensitive especially in some of the red wavelengths. And also, how well does that PMT respond to different wavelengths? So ranking dye brightness and fluorochrome response on individual flow cytometers, they are different. Because the PMTs have got different responses to different wavelengths. Your cytometer model, how, what its optical configuration is. How much noise do you actually have in your detector will play a part as well. Do you have good, clean power, or is there, is your lab reasonably noisy? All these things will have a part. We know that some dyes are brighter than others, but because of the variation in instrument to instrument, the collection optics, you will get a different response. And also, something that is important is how well you look after your instrument, and how clean the optics are, and if things do start to degrade, it really is a good idea to replace some of your bandpass filters. So what can we do to improve how we select different markers associated with fluorochromes when designing our panels? Ranking fluorochromes for individual instruments, which is what we've done, is a good start. And what we've done here on one of our fluorescers is that we've grouped, the different fluorochromes into, very high, high, moderate, low to very low. And these groups of high or low fluorochrome, brightness are going to be different for each instrument. So the fluorochromes that are going to be very high on one instrument may not be the same on another instrument. So people need to be aware of that if they're going to be staining a certain panel using, that they've designed on one of the instruments. They may not get as good a result on some of the other instruments. Things to consider when you are going to be designing, multicolour panels. You really need to determine, define how good your dye brightness is on an instrument, and try and avoid any fluorochromes that do spill over. You don't, the less overlap there is, the better your resolution. Another suggestion is, if some of the dyes are overlapping, then try and use markers for those overlapping fluorochromes that don't co-express, something like, T and B cells. And try and choose bright dyes for some of the low expression markers, especially the fluorochromes that don't have a lot of spillover. Understand the biology of your antigen, or what you're what the question is you're asking with your experiment. Define the dye brightness for each analyzer, and then also consider the spreading error of that dye on the instrument that you're going to be using. Also, something you need to take into consideration is, some of the markers you're looking at transient. Does something biological have to happen for that marker to be expressed? And in those cases, really try and use fluorochromes that are bright or have very minimal spillover. Know your flow cytometer. So what do we need to do? Well, we continually update and educate our facility users about dye complexity and panel design. We emphasize the individual optical characteristics within the facility, and we'll sit down with our users and help tailor their panel, to the instrument of their choice. We've ranked all our dyes and use some of the features in software that will allow you to calculate the spread of your fluorochromes for that particular cytometer, and provide this as a really good resource for facility users, to help guide them to better panel design and some of the more complex panels that we've been doing for the clinical trials that we do here. We aim to give the best advice and service, because we are a service facility, to our users, and we will modify our instruments as new dyes come on the market to help further improve the service to our users. And I just wanted to thank the rest of the gang here. It was a pretty major day or days, multiple days when we did this work. Every single person had an instrument, and they basically almost spent the whole day on each instrument, running all the different heaters and all the different fluorochromes, just to get all this data together. So big effort by everyone, and Thank you. So any questions?
Thank you, Grace, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Grace Hajnowski will answer as many questions as time permits. The first question is, how important is titration of each antibody conjugate used?
It's very important. There's, as we saw with the staining index, having the highest titer doesn't always give you the best separation of the cells of interest. You may actually start to increase the background a little bit more if you do have too high a concentration. And then again, if you don't have enough, if your concentration of your antibody is too low, you're not going to get a very good signal either. So it's crucial that you do titer your antibody, before you start doing any experiments.
Thank you. You mentioned doing an antibody titer using comp beads. How does this compare to titration on the cells, and what are the advantages and disadvantages of doing titration using comp beads?
So the advantages are that you don't need to collect, large volumes of blood for all the fluorochromes. You can imagine, 61 different antibodies with, numerous titers. And we wanted to make sure we had, the same sample. So you do need to collect a lot of blood, which, some volunteers are happy to give, but, after a while. But with comp beads, they're if you get the same lot number, they're constant, the concentration is the same. The only disadvantage with using comp beads, they do have a lot more receptors on the surface. So, the amount of antibody that will bind to them is always a lot higher. So you will end up using a lot more antibody. So what we did do, we really tried to make sure that the concentration that we used was constant for every comp bead. So there's pluses and minuses for both, but comp beads, they're there all the time, and there isn't as much effort in preparing them as what you need to put in when you are using peripheral blood.
Thank you. Okay, your next one is, I like how you classify dye into brightness categories, say dim, moderate, bright, very bright. What about doing this for antigen density information, say low, medium, high density?
Yes, that is important as well. So where, if you do have a high antigen density, you would rank the, where what you expect, or whether you are going to get a high antigen density, and you would use the cells, or you would use the fluorochromes that maybe aren't as bright with some of the antigens that are bright. So, for example, something like CD8, has got a very high antigen density. You could use some of the dimmer dyes on something like CD8, whereas, other markers or that may have a very low antigen density, you would assign some of the brighter dyes. And you would also try and use the dyes that are bright, but also ones that don't give you too much of a spillover. And then the antigens that you're not sure of, or that you're testing for and you don't know what their expression is going to be, or ones where there is a transient expression, you would try and assign dyes to those that don't have too much of a spillover and are reasonably bright as well.
Thank you. Okay, the next one is, can you summarize the instrument characterization studies you recommend doing for any flow cytometer? Does this characterization ever need to be repeated?
So it should be repeated every time. If you have your photomultiplier tubes changed, or the instrument's realigned, you get a new laser. Any of the optical components are changed, it will make a big difference. One of the older instruments that did give us poor responses, we've started to purchase new filters, and we haven't repeated the studies, but we'll once we start doing this again, we'll be able to see a marked difference and just show the difference between what the characteristics were before we replaced a lot of the filters, and then how the instrument behaves after some of the optical components were changed. So realistically, the characteristics will change as soon as you change anything on the instrument, and also over time, as I showed in some of the slides, things will degrade, and the characteristics won't be as good as what they may have been a few years prior.
Thank you. It looks like we have time for one more question. That will be this one. What do you think is the main reason for the differences in the staining index on different instruments for the same dye?
So the main difference could be the laser, depending on some of the yellow-green dyes. If you're exciting them with a yellow-green laser, you are going to get a better response. Some instruments don't have a yellow-green laser. They're using a 488 excitation, which isn't as optimal for those dyes. So you're not actually exciting the dye at its optimal absorption. The other component, which I think is very important, are the actual collection filters. I found that they make a huge difference as to how good a signal you get, depending on the quality of the filter and whether it has degraded, and also to some effect the actual photomultiplier tube. Some just have a little bit better sensitivity, to certain spectral wavelengths.
Well, I would like to once again thank Grace Hajnowski for her presentation. Grace, do you have any final comments?
No, but if any, I can't think of any at the moment, but if anybody, would like to ask any questions, please feel free to contact me. My contact details are on our website, the QMR Berghofer website.
Well, thank you, and I'd like to thank the audience for joining us today and for their interesting questions. And as Grace said, questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through January 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.